Welcome to the Soil CRC's Building Technical Capacity for Improved Soil Management webinar series. Soil Biology Overview. In this video, Dr. Justine Cox from the New South Wales Department of Primary Industries provides a thorough overview of soil biological diversity and function, the good and the bad. A practical guide to understanding, managing and monitoring in the field, including the influences on soil biology by such things as pesticides.
Okay, so I want to talk about understanding soil biology, managing it, and also monitoring, because there's a lot of interest in all of those aspects of soil biology. So I just want to introduce the soil organisms, and we often sort of group organisms by size because it's very convenient, and it also relates to their function a bit. So on this left-hand side is called the microfauna, and then the very small end of town. So the smallest are the bacteria. So right on the left-hand side. So bacteria, you need a very high-powered microscope to see. And then you've got the fungi, which have a big range of sizes. Some you can see with your eyes, some you might need a microscope to see. And you've got groups, the nematodes. They are these worm-like creatures, and they've got a very large range of sizes too. Protozoa, and you've got this rotifer. And so then you get to the groups of organisms called the mesofauna. And that's about 0.2 millimeters to 10 millimeters. So you can see some of these with your eyes. But if you want it close up, you need a microscope. So all of these organisms run around in the soil and the litter. So things like your mites and your centipedes and collembola and isopods, so slaters. And when you get above 10 millimeters, we call that the macrofauna, and that's the larger thing, beetles and the amphipods. And so because they're bigger, they have a bit of a different role in the soil. So I just wanted to give a quick overview of the organisms and how they're sort of classified. But we're very interested in the function of the organisms in the soil and why they're important. And a lot of it in agriculture comes down to their role in decomposition and associated nutrient cycling and carbon storage and turnover. So these organisms, their primary role is to decompose organic matter in the soil. And in that process, they use organic material and they shred it and use up the carbon and then release the organic forms of nutrients like organic nitrogen, organic phosphorus. And then other organisms slowly convert that to mineral forms where plants then can take that up. And that's what happens in nature, in forests and grasslands. And that's how plants get their nutrients. Also in carbon storage and turnover, so organisms require carbon to function for energy. But they also create different types of carbon molecules. And there's this constant dynamic changing of carbon forms in the soil. Carbon dioxide is released from the soil, and also carbon is inserted into the soil through the roots. So there's very, very dynamic carbon stored in the soil and the roots. And also organisms are really responsible for rearranging soil particles and structure. So they can create pores, holes, and help bind soil particles together. They're involved in suppression of soil-borne diseases. If you've got a very diverse and active group of soil organisms, then they can outcompete or they can produce other molecules to prevent build-up of disease organisms. And there's a couple of different methods and mechanisms where they do this. Also, some of them can transform potentially harmful elements and compounds. So some of the herbicides, fungicides and insecticides can be converted into less harmful elements. So here I've got a picture of down in the bottom of the fungal threads where you can very clearly see the white threads. So let's take you through the organisms again, and specifically about the roles each of the groups have. So the microbes, which are the bacteria and the fungi. So bacteria decompose organic matter. So they sit on plant roots and organic matter and old rotting insects and plant roots, and they decompose what they're attached to and release plant available nutrients. So they do that via enzymes. So they release enzymes, or they have enzymes inside their cells to catalyze that conversion from organic nutrients to inorganic. And while they do this, they also secrete sticky proteins and gums which stick soil particles together, and they form aggregates. So they have very important roles in the soil. Fungi, with the larger organisms, they're long strand and thin. They're hyphal threads. That's what the threads are called. They also decompose organic matter and also lignin. So their role is to decompose sort of woody material, and also they can decompose lignin in wood. So they can also penetrate into larger pieces of organic matter and also inside living plant roots. So they wrap their threads around these aggregates, and they also then improve soil structure. They can hold the aggregates together and make them more stable whenever the soil gets wet. And there's a lot of research looking at the mutually beneficial relationships with plants. So there's fungi and plants go together in many, many plant species, and there's so many interactions to discover here. Protozoa then are the single cell organism, but they're mobile, and so they run around in a thin water film, and they eat bacteria, fungi, and organic matter. So these are quite an important group in the food web. And so let's go up to the larger organisms, the microfauna and then the mesofauna. So nematodes. Nematodes are a tiny sort of worm-like organism. You can hardly see them, but they're the most diverse group on the planet. There's 40,000 species that we know of, and there's probably more. 80% of the individual animals on Earth are nematodes, and they are everywhere, and they're mostly beneficial. Although sometimes you hear very bad press about nematodes, that they do cause a lot of diseases, but the majority of them are very essential parts of the soil food web. So nematodes eat a lot of things. Like individual different nematode groups are very specific in what they eat, but there's nematodes that eat bacteria, nematodes that eat fungus, nematodes that just eat protozoans, nematodes that actually eat roots, nematodes that eat other nematodes, so they're carnivorous. And then there's some nematodes that just eat everything. It's garbage guts. And so there's a lot of work being done, look on the community sort of structure of nematodes, which can tell you a lot about the system that they're in. The larger organisms like the springtails, the mites, the slaters, the diplura, amphipods, all the tiny little things you can see running around, they have an important role in shredding leaf litter, and so that increases the area then for microbial decomposition. And then they're predators, so they eat the organisms below them in the hierarchy. So they are predators and regulators then of microbial populations. So microbes just don't grow and grow and grow forever, because there'll be predators out there to eat them. So if we want to know where they are, most soil organisms are really active in the top 30 centimetres. And most microbes live in a thin film of water around soil particles or plant roots. So they're actually almost aquatic organisms. If you have a look at this diagram of this sort of plant and root system, if we have a look at the middle sort of picture, that's a root. And so all the organisms are very much associated around the root. And there's like almost a gel or sort of aquatic sort of environment around the root. And that area then is called the rhizosphere. On the right, you've got some fungi sort of around on the outside of the root, or also entering into the cells and proliferating inside of the root. And some of these will be beneficial and some of these might be harmful. So what regulates soil organisms and their activity? Because if we want to know about them and we wanted to see if we can manage them, we need to know what regulates them. It's really vital that you know your soil type, or especially texture, because it's very important in the activity that you should expect in a soil. So from the extremes, like from a very sand will be very different to a clay. And that's because of the size of the particles. So the sands have really large particles compared to clay, which are very small particles. So the spaces in between the particles is like the pore size, and then how much water that allows through the soil. And then the penetration resistance, so how hard it is for, say, a root to get through the particles. So texture is really important and will govern a lot of the activity. Moisture is critical. These organisms live in water and live in moisture. So if it's very dry, very, very dry, then you can't expect organisms to be active and abundant. But also within a soil type, then season and climate. So is it the wet season of your year? Is it in the longer term, is it a El Nino, El La Nina or El Nino year? And what parts of Australia are you in? So if you're in hot and wet or cold and wet, that will have a big difference on the activity. And organic matter. So that's food. So it's essentially food, housing, and moisture. So carbon, nitrogen, phosphorus, sulfur, all of the elements in organic matter. So if you don't have any food, then you won't have any organisms. And it's not just carbon. There is a whole complex range of carbon molecules that organisms and diverse organisms will require, and also the rate of replenishment. So if you've got a lot of organic matter and then you've got organisms, then they will slowly convert that organic matter to carbon dioxide and reduce the amounts of carbon in the soil. So is there any replenishment of that carbon? Otherwise, we will lose it. So it could be added through just plant growth, because when plants grow, they add their biomass, they inject a lot of carbon into the soil through their roots. Or if there might even be added organic matter or mulch or manures to the surface and then replenished that way. Oxygen, they're an aerobic organism like we are, so aeration. So most organisms are aerobic, they require oxygen. Some organisms can live in no oxygen, and they are very different to the aerobic organisms, and they use different mechanisms to get their energy and their gas reserves. So you can imagine a very compact soil will have very little oxygen in it, so that might affect the amount of microbes that can survive and thrive in that environment. And we've also got temperature. Temperature is very critical in the organism's activity, and so that's related, say, to season, surface exposure. If you've got a very sort of hot environment and you've got a lot of surface soil exposed, then that's going to be a zone where you have no activity at all. Daily fluctuations, you might have a soil that fluctuates a lot in temperature, and so that might not be conducive to a lot of activity. So that again depends where you are in Australia on temperature. So they're more active when it's warmer up to a point, and we'll get an area where it's too cold, then they won't be very active at all. pH is critical. In general, this is a very big generalisation, you want your soil between 5 and 9 for organisms. Bacteria sort of like around neutral. Fungi can tolerate acidic environments. But you will have sort of crop requirements, which will dictate what pH you're aiming for as well. But you don't want to go too extreme for pH. Disturbance. So there's quite a lot of elements about disturbance, like the frequency. If you have a disturbance and it's done once, then it might not have a really big effect. But if you are doing a disturbance to the soil and it's like every month, then that's going to select a particular type of organism that can tolerate that disturbance. And so say if you've got a lot of fungal threads that get exposed month after month, then they're not going to be able to survive in the long term. The magnitude, so if you have a really big disturbance, so like you invert a soil, pick it up and turn it upside down, that's going to have a really big impact on the organisms. And the depth, so if you're just doing something at the surface, that might just affect the surface organisms, and they might be able to be quite happy underneath there. So these are the kind of things to start thinking about. So plant chemicals. So we're talking about those root carbon and other sort of molecules that the plant creates and sends out through the roots to provide food for organisms. And also there are a lot of signaling molecules in that chemistry, and they can sort of regulate and sort of select some organisms. And there's a lot of research looking at how specific those signaling molecules are. Are they just saying, come and get some food, or are they trying to attract a particular type of group of organisms? So I'm just reinforcing that activity is really related to moisture and soil types. So on this graph, there's on the bottom axis is moisture proportion of water holding capacity. So here's 100%. And this is on the vertical axis is the amount of carbon dioxide that is captured, and that's sort of respiration. So that's the organism sort of breathing, which would give you a measure of how active they are. So here's this end on the left is dry and on the right is wet. So a lot of these lines are curved so that they actually are most active in the middle. So not too dry, not too hot, a bit of like Goldilocks, like your best in the middle. And so, but you can see a lot of differences in these lines. So those different soils will have a different response. So some of these will be sands and some of these will be clays, and they will have a different activity. And microbial activity is also controlled by the amount of available soil carbon. So this graph shows on the x-axis, this horizontal axis, the amount of available organic carbon. And on the y-axis is a index used about activity of microbes. And so with more carbon is more activity. So they really do go to town when there's a lot of food out. So it's like a buffet. You know, if there's lots of food around, everyone's going to be really happy eating all the things they like. So knowing that, is it possible to manage the soil and manage your soil biology? Well, let's have a think. So we know how important moisture is. So what can we do to help them maintain a good moisture? If you can soil cover where possible, and they're really going to depend on your enterprise, whether you're cropping, pasture or horticulture. So if you've got a lot of bare exposed soil, you'll have evaporation at the surface. So if there's a possibility of getting some soil cover, that will help maintain moisture retention. It may be possible to add mulch to the soil and cover the soil completely. You can use cover crops. And if you use organic amendments and incorporate them into the soil, they, depending on which ones, have a potential to hold more moisture in the soil. And you can irrigate. So we're just trying to get a really good, as much moisture held in the soil as possible. And it really comes down to organic matter quality and quantity. So if we can increase plant biomass, that will give us more organic matter in the root zone. and when the roots die and then be converted back to nutrients, and those root exudates that are sent into the soil. Cover cropping, you can retain stubble and trash, add mulch, add compost, add manure. And so we're dealing with the quantity here, but quality is also important. So if you have a diverse inputs of organic matter, so they're really sort of long-term, short-term, and then medium-term, and of different types, you'll have a diverse amount of organic matter, and that will directly then encourage a diverse group of organisms to come to that area and thrive. And we'll talk about diversity a bit more. Converting cropping to pasture has been shown to increase organic matter. And here we go, diverse plant species will essentially give you diverse microbe species. Rotation, so if you add a few rotations, that will introduce different plant species, then the different organisms will develop. Stocking rates, you'll have additions and organic matter, and even if you just want to reduce losses, that will maintain a high organic matter. So if you've in an area where you lose a lot of soil in soil erosion, if we reduce those losses, then we're going to improve our organic matter content. Aeration. It's really tricky and really hard to get air into soil once it's compacted and closed up with little pores. So we've got a few options. Deep-rooted plants have been used to try and get air into an uncompact soil, and that's really hard because if it's compacted, it's really tricky to get roots to down there. So there's some research trying to find some effective deep-rooted plants that will grow. You might want to, if you've got a no-till system, maybe an occasional tillage is something that's will be beneficial. There are some mechanical aerators for this very sort of situation. They might have a core sort of function, or a punch hole kind of function, or a like a very thin line that they can rip. Organic amendments. There's some research trying to actually look at sort of a ripping and organic matter combo to get organic matter down to depth, which will help with aeration. Also, preventing compaction in the first place is really important. So no heavy equipment on wet clay soil you will then avoid the compaction issue. Temperature. Sometimes we can't do too much about it, but if we can cover the soil surface. We can do that with either plants or in mulch. In some horticulture, you try different coloured plastics to change the temperature underneath. pH, so if it's getting very acid, we might have to use some lime to increase the pH. If it's very alkaline, there's sulfur and other methods. Again, organic amendments are useful to maintain a good pH. If we can choose a less acidifying nitrogen fertiliser, that might help in the long run. But it also really depends on your crop system and what pH your plants prefer, and what method you have of changing the pH. It's very difficult to change pH. And so some systems sort of you'll get a little bit more bacteria, and some systems you'll get more fungi, depending, say, if it's a tree crop system like macadamias where I work, will be quite a bit of fungi in the system. And again, that disturbance. So depending on your crop system, you might want a minimum or occasional tillage, maybe an option. We could add soil organisms, and there are some examples where this might be useful and essential in some organisms. So rhizobium are inoculants that will be essential for legumes, because these are the bacteria that provide the nitrogen. And so these can be very specific to a particular pasture species, or some of them will be generic. So, but they're essential. mycorrhiza, which are a fungi, and again, some of these are specific and some of these are general, but mycorrhizae provide a benefit to the plant. They get them more phosphorus, more water, and some mycorrhizae then get food from the plant to the mycorrhizae, so it's a bit of a beneficial relationship. And there are a lot of biological products in the market, and there are so many and we don't know all about them, so it's a bit hard to sort of judge their efficacy. So you need to know that they have research behind them to show that they are useful. So here's a picture of our nodulation of our pasture legume. So there's an example of a trichoderma product for inoculating a soil for protecting against other diseases. I think that might be onions. And so also then farm chemicals have herbicides and insecticides and fungicides. There are so many chemicals out there. We don't know the effects of each one of those on the biological universe, but I have several colleagues that are working on this, looking at effects of these different, at the moment, herbicides. And I'll give you an example in this talk at the end. Okay, so now we know there's some options to manage them. How do we know if it's having an effect? So we want to know how to monitor. But before we go, I want to monitor my soil biology, we need to ask a question or several questions. So what's your goal? Do we just want to monitor over time and see what's happening? Do we want to use it to diagnose a problem? Is there a problem that you maybe think that soil biology has an input into? Do you want to compare, do some comparisons for some of your sites on farm? Is there, just you want to use the information for a decision support tool? So we really got to get really specific about what we were trying to find with using soil biology. And then what's your time frame? Is it something you need to know this week? Is it talking about this season, this year? Are we just going to monitor continuously? So there's a lot of limitations in monitoring soil biology, isn't there? There are no standard methods. I mean, we've used methods in our research, but other groups in the country will use different methods. And so there's no sort of universal method that, like in Australia, you would get, except for some of the disease assessments. But generally, there are no numbers. There's no guidelines on this is low, this is high, and this is good. And often there's no direct relationship with plant productivity. So you might have the plants going gangbusters, but the biological habitat is not that interesting or diverse. So there are a lot of limitations, and we are exploring and expanding on our knowledge in soil biology. And the CRC projects are doing that in spades. Here I just wanted to add that soil biology is important for a lot of soil health monitoring that growers and grower groups have really sort of led the way in. And here's the Northern River Soil Health Card. I was involved in developing that with a whole range of colleagues and the Soil Care group. And so they wanted some ways of monitoring things that were not just the chemistry lab results. And then the Victorian Soil Health Guide has a component of soil biology in there. So I just wanted to throw those up as useful things to go and have a look at. But this is the guts of what you wanted to know, is how do you test soil biology? And I've separated into in the field and in the lab. In the field, we can count things that we can see. We need to take good samples or good methods, but we can count earthworms in a known volume. We can count mites and ants and those Calimbula and dung beetles. And we also can count the percent nodulation of legumes. So like, how many nodules on this plant are red, and we know that they're active. So that's one way we can test. And we want to know, well, what about the activity? So we really don't need to know who's there, but is there activity and the function of the soil happening? And there's a lot, and we're really focusing on decomposition because that's our main action that we want to sort of see is happening. And there are a couple of different ways you can do that in the field. One is using cotton strips, which is just unbleached calico placed in the soil. I'll show you a bit more. And then the organisms decompose that, and you can take that out after time and have a look. Which is similar to Soil Your Undies program, which is getting cotton men's underwear and digging that into the soil and digging that out and taking a picture. Litter bags, which you place the litter in the soil in a bag with a mesh screen so that organisms can come in and out. And then there's the tea bag index, which uses a standard tea bag. There's very specific tea bag, two tea bags. One's rooibos and one's like a normal black tea. And then you see how much of the tea leaves are decomposed. And activity, then the product of decomposition is respiration, so the release of carbon dioxide. And there's a commercial test that measures that carbon dioxide, but really easily. It just turns a gel paddle a different color, and then you can determine how much carbon dioxide is coming out. Indirectly, we can look at the percent ground cover, because that will have an indirect component to the soil biology. We can have a look at root volume and distribution. That's a bit tricky. You need to get a really big spade if you've got a big plant. And there's a physical Emerson dispersion test, which actually is related to soil biological activity. If you place an aggregate in water and you see if it disperses, this is slight and then moderate and severe. If it just collapses, then you know that there's no glues or those from that bacteria and fungi holding those aggregates together. So you've got a very good, high active, really biologically active soil. Then you might often see nothing happen when you add water. It just holds its shape and its structure.
Justine, would a slaking test also give the similar?
It's the same, yeah. It's a slaking test.
So yeah. So you might not get dispersion, but you might get slaking, and so even slaking is an indicator that it's not really being held together by anything.
Yeah. Great, yep, because the biology is related to the physical structure of the soil. So here's a few pictures to show what I was talking about. So the top left one is calico strips. And you insert that vertically into the soil with some coming out so that you get the soil ground level and above and below. And then that will decompose over time and you'll get losses in the calico. You can pull that out and then have a look. You can either just use it as a visual tool to show the amount of decomposition. Researchers actually then test the calico to see the force required to break that strip, and that is, then you get a number for that, and then we can get then some quantitative data. The other way I've actually used this is actually take a picture, and then there's a free software that just so this will have lots of holes in it, and if you take a picture on something contrasting, it then can calculate the area. So if you have a lot of decomposition, a lot of area lost, it will calculate that. And so underneath that is Soil Your Undies. So you get these white cotton undies, and you also put them in the soil and you come out and they're all degraded. And so There's a global campaign around the world where farmers are encouraged to soil your undies and take a picture and upload it to the global website. And Oliver Knox and UNE at Armidale is really coordinating a lot of this in Australia. The top right is that Solvita respiration test, and I use that a bit. It is, you put soil in these jars that they give you, and you put this paddle in there, and you leave it a certain amount of time, and the paddle, this purple here, will turn a different colour. And that will be related to how much carbon dioxide is evolved from that time. And that's really simple. And you get a 3, and that will tell you, what that means. It doesn't, in itself, it doesn't actually mean a lot, but if you're comparing things, you can then compare things very similarly. And these are the 2 tea bags for the tea bag index. So they're a Lipton Rooibos and a green tea. So unfortunately, you have to get them from the Netherlands, but I've got multiple packs. So it's if people are interested, I can let them know more about that. That's backed up from researchers. And that's also a global repository of data and engagement. So in the lab, we can count numbers or presence of organisms, and we can do that in bacteria and fungi. We can do that with plate counts, which grow them on agar or looking down the microscope, but more popular and more encompassing is DNA and other molecular methods. You get all of the DNA of everything in the soil. You can look at nematodes under the microscope or by molecular methods. The percent root infection of mycorrhizae, so how much of the root is infected with mycorrhizae. Microbial biomass carbon, so how much of the carbon in the soil is due to microbes. So that's a good a tool to work out the percentage of the carbon is microbial. And there's quite a number of range of things to look at activity and function. So looking at the enzymes themselves, which actually is the conversion from organic to inorganic. And they can be very specific, like a single enzyme transformation, or general. So if we look at beta glucosidase, it just looks at the glucose conversion. But if you look, there's this test called fluorescein diacetate, looks at all sort of general decomposition. We can do decomposition rate in the lab as well. We can get soil samples and put cotton strip tests in the soil in lab. And we can also measure respiration in the lab. Potentially mineralizable nitrogen. So bring some soil back and estimate how much of the nitrogen will be mineralized under controlled conditions. Then we can look at diversity. There's a lot of testing going on looking at diversity of microbes, and that's a lot of the DNA tests using different parts of the genetic information of all the organisms in a soil sample. We can look at the community analysis, so especially nematodes. If we know what proportion of the nematodes are, if they're all of the nematodes in the sample were like bacterial feeders, then we can tell us something about the soil in that we'll have a lot of bacteria, so the composition of the nematodes will tell us a lot about the habitat. Microbial function, and so looking at the carbon use. So this is MicroResp, which is a tool that our colleagues use here to estimate the diverse use of different carbon molecules. And so we can separate all of the different molecules and see the diverse sort of a pattern And then there's the genetic function. So going right just to the genes and looking at whether we have variation in all the genes responsible for all of these conversions, and that's called metagenomics. There are other indirect ways to look at soil biology, like looking at the carbon fraction. So there's the total organic carbon, there's labile carbon, there's the short, medium and long-term sort of stable carbon. We can look at all of those. We can look at disease organisms, so the predictor B test. And we can grow plants in what's on this picture on the bottom of the rhizo box and look at root studies and see what's happening in a controlled environment. So just quickly, an example of some research that colleagues are doing. This is Mick Rose and Terry Rose and Lucas Van Zwieten. They compared the effects of glyphosate and Roundup. So Roundup is just glyphosate and a surfactant. And so they use substrate-induced respiration, so that micro-resp test, to look at respiration and enzyme activities that are responsible for the carbon, nitrogen, sulfur and phosphorus cycling. And they looked at 3 soils in the lab over 27 days. And what they found at label rates, there wasn't any really effect on either respiration or the enzymes. So no evidence there of action. But at high rates, the sandy soil was actually affected compared to the other soil types, which will be more of a clay loam or a not sandy. They found that enzymes were not as sensitive as respiration. So that's helping us determine what tests are going to be more useful. So if they're sensitive, we might be able to pick up effects more easily. So that micro-resp test is actually coming to be very useful. And they found that Roundup affected soil function more than glyphosate. So the Roundup has that surfactant, and so that component was actually more detrimental than the glyphosate. And one last one is effect of herbicides on soil function. So Mick Rose compared 7 herbicides and 1 fungicide. They're all of the chemicals there. I'm not going to try and say all of those. And they measured 8 enzymes, the mineral nitrogen, to see what effect it would have on 5 soils at the recommended rate, and then 5 times the rate. And they found that all herbicides at recommended rate had little effect except atrazine. Atrazine did affect the soil function. So at 5 times the rate of the metal sulfuron, or the 2,4-D, they did find that caused alterations to the mineral nitrogen levels. So if those rates were, accidentally put on higher or they accumulated over time, then there might be some changes that we can pick up. And again, nitrogen transformations were more sensitive than enzyme assays. So we're honing the different tests, we'll be able to work out which ones are more sensitive. So just to summarise, so soil biology testing is complex and there's no optimum or minimax numbers for each crop, climate, soil type, because imagine Australia is so diverse. But they have value in comparisons or monitoring over time or answering specific questions. So build your own monitoring schedule and then you'll build your own data set. And it's likely that a suite of mixed indicators will emerge as useful for management decisions. It might be a combination of something which tells you about numbers, something that tells you a bit about activity, and something tells you a bit about diversity. So keep involved, keep on the cutting edge, and keep getting dirty. And I'll just end you with some further reading. There's some amazing things going on. Yeah, you have to do your own research on your own property. Unfortunately, there's so little known and we don't know how it's going to impact everyone's particular crop or soil or climate. So essentially, if you're really interested and you think that there's valid reasons for it, then you set up your own trial.
Yeah. And I
I
think
might just jump in there. Sorry, Justine.
Cameron.
A colleague of mine, Anders Claessens, is a mycologist, and we're currently doing a project with Sumitomo, who are a large chemical company, but they're trying to branch into microbial inoculants, and they're doing some work looking at arbuscular mycorrhizal fungi as an inoculant and how they affect growth rates on various crops in Australia. So we're doing that in partnership with the Northern Growers Alliance. And what we've found so far is that there's a whole bunch of different fungi that you find in the soil. And so if you inoculate your soils with those fungi, people are going, that's great, look, we found them in the soil, there they are. But not all of those end up in the roots where they're actually needed. So just because you're inoculating your soils and you're increasing that composition doesn't necessarily mean that that's relating to the function or what's needed in that plant. So microbial inoculants are an incredibly new space. And as Justine said, unfortunately, there's no blanket guideline yet. But at the time, the research is currently being undertaken. So.
Thank you for watching. If you found this information useful, there are many more related projects to discover at our website. Please like this video and subscribe to our YouTube channel for more Soil CRC webinars, project updates and conference presentations. This webinar series was produced by the Soil CRC, jointly funded through the Australian Government's National Landcare program, to build the technical capacity of natural resource management agencies, Landcare and grower groups to deliver soil health information to farmers for improved soil management. For more information about our projects, visit soilcrc.com.au.